a little salty about active transport, this is probably why. Hi biology friends, it's Sarah, your other biology professor, and let's talk about the sodium-potassium pump. So again, we're in the world of active transport. That tells us a few things. First, we're transporting something against its concentration gradient. That means we're moving it from an area of low concentration of that substance to an area of high concentration. We're concentrating it, stockpiling it. Second, we're going to need a protein to do some work. And finally, that's going to burn some energy. We're working with two ions here, sodium and potassium. Because they're charged, ions cross the cell membrane through a protein. And for active transport, we're using the sodium-potassium pump. In one cycle of pumping, 3 sodium ions are pumped to the outside of the cell, and 2 potassium ions are pumped to the inside of the cell. Let's see how this works and why the cell does this. In this diagram, we've got our cell membrane and the sodium-potassium pump. The space below the membrane is the inside of the cell, the cytoplasm, where the potassium concentration is much higher than it is outside the cell. And the space above the membrane is the outside of the cell, the extracellular environment, where the sodium concentration is much higher than it is inside the cell. High potassium inside the cell, high sodium outside the cell. To begin our cycle, the pump faces inward toward the cytoplasm. In this setup, it's got a high affinity for sodium. It binds strongly. 3 sodium ions from the cytoplasm bind sites on the sodium-potassium pump. The pump is phosphorylated by ATP hydrolysis. ATP binds the pump, which works as an enzyme, pops off a phosphate group, ATP becomes ADP, and that phosphate is stuck onto the pump. This releases energy. And it causes a conformational change or a shape change in our pump. The pump shifts to an outward-facing conformation that has a lower affinity for sodium. So the sodium ions are released to the outside. They've been moved from an area of low concentration of sodium to high concentration of sodium. This reveals 2 binding sites for potassium ions. 2 potassium ions bind the pump. That causes dephosphorylation of the pump. The phosphate group is removed. So now the pump returns to its original inward-facing conformation. The 2 potassium ions are released into the cytoplasm, and again, 3 sodium binding sites are exposed. So in each cycle, we pump out 3 positive charges and bring in 2 positive charges. That's a net export of 1 positive charge to the outside per cycle. That means that the inside, the cytoplasmic side of the membrane, is more negative than the extracellular side. We've got an electrochemical gradient. So not just a concentration difference, but a charge difference across the membrane. But why? Well, among other reasons, this establishes the resting potential of the cell, the difference in charge across the plasma membrane. Cells can use this to do things like send a nerve impulse or contract muscle. Setting up this gradient is also important for transport. In regions like your gut, cells use the sodium gradient maintained by sodium-potassium pumps to help absorb glucose using another protein transporter. And the sodium-potassium pump helps your cells stay at the appropriate volume by maintaining ion concentrations to prevent osmosis-related swelling. In fact, they stay so busy that sodium-potassium pumps use about 1/3 of our ATP, and even more in cells like neurons. Remember, it's like a pumpkin. We pump K in. That means sodium goes out. There's tons of good memory tricks for the sodium-potassium pump out there, so if you know one, drop it in the comments. Otherwise, give me a like if you learned something about this sodium-potassium pump. Subscribe for more biology content from me. Check out my website for tutoring resources, and I will see you next time. Bye.